Imagine this scenario. You're watching an animal, maybe a llama on a farm, or a deer just grazing peacefully, and suddenly something's clearly wrong.
Yeah.
They're stumbling, maybe staring blankly, sometimes even having seizures. It's really alarming, this neurological puzzle that just seems to pop up.
Mm-hmm.
And what if I told you the key often lies in something really basic, like what they're eating?
It's surprisingly common, yeah.
So today, we're doing a deep dive into Polioencephalomalacia. PEM for short, thankfully.
Definitely easier to say.
It is, but that long name, it literally means softening of the brain's gray matter, which is a pretty stark description.
It paints a picture. Certainly, it's a neurological syndrome affecting unique animals like camelids.
Yeah.
Your llamas and alpacas and cervids, like deer and elk.
Okay, so our mission today is to unpack the sometimes surprising causes behind this, how it looks, why some animals are more vulnerable than others.
Right? And crucially, how quick thinking, or even better, good prevention can make a huge difference.
Yeah, get ready for maybe some aha moments about how metabolism, the environment, even how we manage animals can play a role.
It's fascinating stuff.
And this isn't just, an animal health issue. It's a really interesting look at how delicate our internal systems are, how small changes can have big consequences. You'll see how critical thinking, looking at the whole picture, is key.
Absolutely, whether it's animal health or really any complex problem.
So PEM, you said it's a syndrome, not quite a single disease.
That's right. Think of it as a common endpoint. Different things can lead to the same pattern of brain damage, specifically neuronal death. Fundamentally, PEM is about the brain being starved of energy.
Starved of energy.
Yeah, leading to what we call cerebrocortical necrosis, CCN. That's the actual death of brain tissue, the softening in the name.
So if the brain's starving, what's the critical fuel that's missing or blocked?
Good question. The big player here is thiamine, vitamin B1.
Ah, okay. B1.
Yeah. The brain uses a massive amount of energy, and it runs almost entirely on glucose. Thiamine is like a crucial helper, a coenzyme, needed to actually process that glucose.
Right.
It helps convert glucose into ATP, which is the cell's energy currency, through pathways like the Krebs cycle.
So without thiamine.
Without enough active thiamine, those energy pathways just sputter and stop. The brain can't make the ATP it desperately needs.
Okay.
That leads to brain cells swelling up, problems with neurotransmitters, basically widespread energy failure, and then cell death. It's like the fuel pump feeling in a really high-performance engine. The engine just starves.
That's a really clear analogy. And if you could, look at the brain itself afterwards, what would this energy crisis look like?
Well, at necropsy, the post-mortem exam, the brain often looks swollen. The normal folds, the gyri, might look flattened out.
Okay.
You might see some yellowish discoloration, especially in the cortex. And here's something really distinctive. Those damaged areas, they actually glow. They autofluoresce under UV light.
Wow, really? That's quite a specific sign.
It is. Very useful diagnostically.
Mm-hmm.
Then, under the microscope, we see what's called laminar cortical necrosis. That means specific layers of the cortex show neurons dying off. They look shrunken, damaged. It confirms that widespread cell death from the energy crisis.
Okay, so thiamine is absolutely vital. Without it, the brain just can't function. But what causes this shortage? Is it just, not enough B1 in their food, or is it more complex?
It's almost always more complex than just not enough B1 in the diet. Historically, yeah, PM was pretty much thought of as just thiamine deficiency.
Right.
Because in healthy adult ruminants and pseudo-ruminants like llamas, the microbes in their gut, the rumen or the C1 compartment, usually make all the thiamine they need.
Ah, so their own gut bacteria make it for them.
Exactly. But lots of things can disrupt that process.
Okay, so it's about their internal ecosystem, not just the feed itself.
Precisely. A really common trigger is a sudden diet change, especially switching to feed that's high in carbohydrates or concentrates, but low in roughage.
Like feeding too much grain suddenly.
Yeah, exactly. That can cause ruminal acidosis, the rumen pH drops. And that acidic environment, it favors certain bacteria that actually produce thiaminase.
Thiaminase? What's that?
It's an enzyme that destroys thiamine, breaks it down. So bacteria like Bacillus thiaminolyticus start to thrive, and they actively sabotage the animal's thiamine supply.
Wow, so the bacteria are literally eating the vitamin before the animal can get it.
That's a good way to put it, yeah. They're destroying it.
Okay, what else can mess with thiamine levels?
Well, sometimes animals eat plants that naturally contain thiaminase, things like bracken fern or horsetail.
Yeah.
That introduces the enzyme directly.
Right. Makes sense.
And then there's something called thiamine antagonists. This is a really key point, often missed.
Antagonists. So they work against thiamine.
Exactly. They're compounds that look chemically similar to thiamine, enough to sort of trick the body, but they block its function instead of helping.
Okay, like what?
The big one, clinically, is amprolium. It's a common drug used to treat coccidiosis, a parasitic infection.
Oh, wait, so a medicine, something meant to help.
Yes. If it's not managed correctly, amprolium can actually cause PEM by creating a functional thiamine deficiency. We call that an iatrogenic risk treatment induced. And it's a really significant concern, especially in camelids, because they often need treatment for coccidia.
That is. Wow. That's a huge thing for owners and vets to know.
Yeah.
So a treatment could lead to this severe brain issue. That's one major cause pathway. But you mentioned there's another big one, more recognized recently, something about sulfur.
Absolutely. Understanding the role of sulfur has really shifted how we approach PM. It's become a major focus.
Where does the excess sulfur come from?
Various places. It can be high in certain feeds, especially some agricultural byproducts like distillers' grains or corn gluten feed.
Things often used in livestock rations.
Exactly. Also, some plants like crucifers, even alfalfa sometimes. And importantly, water sources can sometimes have very high sulfate levels.
Okay, so the animal takes in too much sulfur. How does that lead to brain problems? Is it another thiamine issue or something different?
It's primarily a different mechanism, a direct toxic attack. In the rumen, that oxygen-free environment, microbes convert the excess sulfur into hydrogen sulfide gas, HUS.
Hydrogen sulfide, isn't that the rotten egg gas?
Oh, and it's toxic.
Highly toxic, yes. That gas gets absorbed into the blood and goes straight to the brain. It's a potent cellular poison. It specifically inhibits an enzyme called cytochrome C oxidase.
Okay, what does that enzyme do?
It's absolutely crucial for making energy in the mitochondria, the cell's powerhouses. H2S basically throws a wrench in the works of that energy production line.
So it shuts down the brain's energy factories directly, like putting the wrong fuel in the engine, maybe.
That's a good analogy, too. It causes energy failure that way, leading to cell death. Different path, same devastating result for the brain cells.
But I think you mentioned earlier, sulfur and thiamine issues can actually interact, make things even worse.
Yes, and this is where it gets really complex and, frankly, quite interesting from a biochemical standpoint. High sulfur doesn't just act as a direct toxin.
It also messes with thiamine metabolism in multiple ways. It seems to increase the body's need for thiamine. It can change the rumen environment in ways that reduce the microbe's ability to make thiamine.
Oh, wow.
And it might even increase the destruction of thiamine already present. Plus, sulfur can bind to minerals like magnesium, which is needed to activate thiamine.
So it's like a double whammy. The sulfur poisons the energy system, and it creates a thiamine shortage at the same time.
Exactly. A literal double whammy. Direct toxicity and a secondary induced thiamine deficiency. That complex interplay is why giving injectable thiamine often helps, even if the primary cause seems to be sulfur.
That is incredibly complex.
Mm.
Okay, so when an animal is going through this brain energy crisis, what are the signs we'd actually see? What does PEM look like on the outside?
The clinical signs really reflect that widespread damage to the cerebral cortex, and they tend to progress sometimes very quickly.
What are the early signs?
Early on, you might see things like depression, lethargy, maybe aimless wandering, and a really key sign, cortical blindness.
Cortical blindness, meaning they can't see, but their eyes look okay.
Exactly. Their pupils might still react to light because the eyes and optic nerves are fine. The problem is in the brain's visual processing center, the cortex.
Got it. What else early on?
Muscle tremors are common.
Yeah.
And that characteristic stargazing posture, it's called opisutnos. The head is held way up high, arch back. Also ataxia, just general uncoordinated movement, stumbling.
And if it gets worse?
As it progresses, those signs just get more intense. You might see them pressing their head against walls or fences, grinding their teeth. That's called bruxism.
Okay.
Involuntary eye movements, nystagmus. They can become hypersensitive to touch or sound, hyperesthesia. And eventually, if it's not treated, they go down, they become recumbent.
Unable to stand.
Right. And from there, it can progress to seizures, coma, and sadly, often death. Sometimes you also see the eyes positioned abnormally, a sort of upward and inward deviation called dorsomedial strabismus.
Wow, those are some dramatic and scary signs. Given how fast it can progress, how do vets figure out if it is PE? Is there one specific test?
Well, here's a fascinating twist. The treatment itself is often a key diagnostic tool.
How so?
Giving an injection of thiamine, hydrochloride parenteral thiamine, If you see a rapid, significant improvement in the clinical signs, maybe within just a few hours,
Then it's likely PE.
it's highly suggestive of PA, yes,
Uh,
especially the thiamine responsive form. This therapeutic trial is often the very first thing you do with a live animal showing those signs. It's practical and can be life-saving.
right. You treat to diagnose, essentially, because time is critical. What other clues help?
A good clinical exam, of course. Noting those specific neurological signs that cortical blindness with the stargazing is a classic combo. And getting a thorough history is vital.
Like asking about diet changes.
Exactly. Recent diet shifts, any chance they got into weird plants like bracken fern, feeds known to be high in sulfur, or importantly, whether they've been treated with amprolium recently.
Okay.
Then, for a definitive diagnosis, especially if the animal dies or is euthanized, the postmortem brain exam is key, finding that laminar cortical necrosis.
And seeing that UV glow you mentioned.
And seeing the autofluorescence under UV light, yes. Blood tests usually aren't very specific, maybe just show stress. Analyzing cerebrospinal fluid, CSF, can be helpful mainly to rule out infections.
So we know the causes, the signs, how to approach diagnosis. But you mentioned earlier that different species react differently. Let's talk about camelids, llamas, alpacas. What's their specific vulnerability?
Right, camelids are pseudo-ruminants. Their three-compartment stomach is really well adapted for digesting high-fiber stuff. But
There's a but.
there's a but. This specialization seems to come with what some people call metabolic brittleness, or maybe a hyperacute sensitivity, specifically to thiamine depletion. Meaning, while a cow or a sheep might take weeks to develop PEM after a dietary problem, camelids, particularly alpacas it seems, can show severe signs in just 2 days.
2 days. That's incredibly fast.
It is. It means there's virtually no time to waste if you suspect PEM. Intervention has to be immediate.
And does that tie back to the amprolium risk you mentioned?
Absolutely.
Yeah.
Because they're so sensitive and develop signs so quickly. The risk of PEM when using amprolium is a huge deal in camelids. You must give thiamine alongside it.
Okay.
And because of this rapid onset, we often advise owners, working with their vet, to actually keep injectable thiamine on hand as an emergency first aid measure if they see those early neurological signs.
That makes a lot of sense. Very practical advice. Now, what about cervids deer, elk? You said their situation is almost a paradox.
It really is, and it's a fascinating contrast. Wild deer are actually pretty smart foragers. They have this innate ability to select plants and seem to actively avoid ones with high sulfur concentrations.
So in the wild, they naturally protect themselves.
Largely, yes, it's a natural defense mechanism. But here's the paradox. Put them in a farmed or captive setting.
And that defense goes away.
It's completely nullified. If they're restricted to a specific ration, especially one containing those high sulfur byproducts like distiller's grains, they become incredibly vulnerable to sulfur-induced PEM.
So it becomes a disease caused by how they're managed.
Essentially, yes. A management-induced disease in a species that actually evolved natural defenses against it. This captivity paradox is a huge lesson for anyone farming deer. You have to watch the sulfur, not just protein and energy. Check the feed, check the water.
That's a really stark example of unintended consequences. Now, you mentioned the signs, blindness, stumbling, seizures. They sound severe, but they could potentially point to other diseases too, couldn't they? How do you sort it out?
That's absolutely critical. Differential diagnosis is key because, yes, those signs aren't unique to PEM. You have to rule out other possibilities to treat correctly and also to prevent other animals from getting sick if it's something infectious.
Right. So what are some key things PEM gets confused with?
Well, PEM usually gives you those symmetrical cortical signs affecting both sides of the forebrain equally. Other diseases often look different, like listeriosis, that's bacterial. It typically causes asymmetrical brainstem signs. The head tilt, one side of the face drooping, animals walking in circles. Very different picture.
Okay, brainstem versus cortex. What else, especially for deer?
For deer and elk, chronic wasting disease, CWD, is always a major concern. But CWD is a prion disease. It comes on really slowly, insidiously, over weeks or months. And the main sign is that profound weight loss, the wasting. Again, very different timing and presentation than acute PEM.
Got it. Any others that affect both camelids and cervids or cross species?
Definitely. Equine herpes virus 1, EHV1 encephalitis. That's a risk if camelids or cervids are housed near horses or especially zebras who can carry it. That causes fever, ataxia, head pressing signs that can overlap with PEM.
Okay.
Then there's ryegrass staggers. That's from a toxin in certain grasses. You see muscle tremors, ataxia. But a key feature is that it gets much worse with excitement or stress.
Interesting distinction.
And another big one, especially in areas with white-tailed deer, is meningeal worm. The parasite, Paraphylstrongylis tenuis. This causes progressive spinal cord inflammation myelitis. Often starts with hind limb weakness and paralysis that moves up the body, and it's often asymmetrical. Again, spinal cord, not primarily brain cortex like PEM.
So lots to consider.
Yeah. And notice how many of these cross-species lines, like EHV1 or Listeria, it really highlights the need for a One Health perspective, recognizing animal, human, and environmental health are all linked, especially when you have different species mixing.
Absolutely. So knowing all this, knowing the causes and the lookalikes, how do we actually prevent PM? What are the key strategies?
Prevention really boils down to smart nutritional management tailored for the species. But some general rules apply to everyone.
Right.
Avoiding sudden big diet changes is huge. Always transition gradually over days or weeks. That helps keep the rumen microbes happy and stable. And always ensure enough good quality roughage, fiber. That's fundamental for healthy rumen function.
Okay. And for those super sensitive camelids?
Prophylactic thiamine. That means giving thiamine injections whenever you treat them with amprolium to counteract its effect. Period.
Right. Non-negotiable?
Pretty much. And consistency in their feed source is really important too, to avoid upsetting their delicate digestive balance. And like we said, have that emergency thiamine on hand, ready to go after talking with your vet.
Makes sense. What about the farmed deer and elk with that sulfur risk?
For them, sulfur testing is essential. Mandatory, routine testing of feed ingredients, especially those byproducts, like distillers' grains where levels can vary wildly. And test the water too.
Okay.
Then you formulate the ration carefully. Keep the total dietary sulfur below the maximum tolerable level, which is generally around 0.4% on a dry matter basis. But you have to factor in water contribution too.
Right. Total intake matters.
Exactly. And if you're managing wild populations or planting food plots, try to choose forage plants that are naturally lower in sulfur, kind of working with their natural avoidance behavior.
And what about biosecurity, especially in mixed groups?
Yeah, that's vital for preventing some of those PA mimics. Keep horses, and especially zebras, physically separate from camelids and cervids if possible to reduce EHV1 risk. And manage silage, carefully ensure good fermentation, low pH to prevent listeria growth.
Okay, so let's wrap this up. We've really dug into Polioencephalomalacia, PEM. We've seen it's this complex syndrome driven by either a thiamine problem or sulfur toxicity or sometimes both interacting.
Right, that dual nature is key.
And we've explored how specific animals have unique weak spots, that metabolic brittleness in camelids causing rapid onset.
And the captivity paradox in deer, where management overrides their natural defenses against sulfur.
Yeah. And we've learned how crucial diagnosis is, needing a sharp eye, a good history, sometimes that therapeutic thiamine trial, and knowing the difference from other neurological diseases.
And that swift action, or better yet, proactive prevention through careful diet management, is everything.
This whole deep dive, it really drives home how important nutritional balance is, doesn't it? And how things we might not even think about in feed can cause major problems.
Absolutely. It underscores the interconnectedness of these biological systems and the need for careful observation, critical thinking, and being proactive. That applies whether you're managing animals or really making any complex decision.
So as we think about these hidden factors in diet causing such havoc, here's a final thought for you, our listeners, to maybe mull over. What seemingly small, maybe overlooked detail in your own daily life, your routines, or the systems around you might actually be setting the stage for a much bigger, perhaps unseen, impact down the line?